right, so last time we talked about prokaryotic cells versus eukaryotic cells. So just a quick reminder, prokaryotic cells were like bacteria, and they're smaller and they don't have a nucleus. And then eukaryotic cells were larger and they have a nucleus and they have all kinds of organelles. And that's sort of where we left off last time with a little overview of some of the eukaryotic cell parts we would be talking about. So this is just a diagram to start us off. This is an animal cell, and you can see all the cell parts that we're going to be talking about. You can see the nucleus, you can see the rough and smooth ER, or endoplasmic reticulum, the Golgi apparatus, sometimes called the Golgi bodies, or the Golgi complex as well. Lysosomes we're going to talk about, mitochondria, centrioles I don't think we're really going to hit on right now, plasma membrane, peroxisomes we'll mention briefly, and then the cytoskeleton that sort of holds everything together. So we're going to talk about all of these over the next few days. Now this is a plant cell, looks a little different. One thing plant cells have that animal cells don't, number one, chloroplasts. Animal cells don't have chloroplasts. Another thing is this really big vacuole here called a central vacuole. Animal cells do not have that. They have vacuoles but they're smaller. And plant cells have a cell wall. So we mentioned this yesterday, some of the differences between plant cells and animal cells. They're both eukaryotic, both have a nucleus, but those were some of the differences. So let's start talking a little bit about the different cell parts. So we're going to start with the nucleus and the ribosomes. So the nucleus is where the DNA is located. And what that does, the DNA, remember, you may remember that it's associated with your heredity. But really, what the DNA is directions for all the proteins that your cell is ever going to build. And since most of the important parts of your cell are protein, enzymes, hormones, a lot of the structures that cells produce, all of those things, surface markers on the cell membrane, receptors to pick up information, all of that stuff is proteins. So that's why the nucleus is sort of the control center. Remember, DNA does not actually leave the nucleus. What actually builds protein is messenger RNA at a location called ribosomes. And then the ribosomes are actually built by a part in the nucleus called the nucleolus. So if you have here a cell, and this is your nucleus, this is where your DNA is. Now, your DNA, you can't, when a cell is not dividing, you don't actually see chromosomes. Like, they don't look like little X's like you might have learned about chromosomes. They're actually in a mass, and you should know this, called chromatin. And it's sort of a mix of DNA and protein, and technically these are chromosomes, but they're really spread out and stringy looking because that's how DNA needs to look in order for it to be actively conducting protein synthesis, making proteins. Think of a book. When you go to read a book, you have to open it, right? So in order for DNA's instructions to be read by mRNA, which would make a copy of the information and then take it to the ribosome, so mRNA would come in and get built. It would copy a little section of DNA, turn it into something called mRNA. mRNA has the ability to leave the nucleus. The nucleus has these little nuclear pores, and the mRNA can go out. And then the mRNA can then go to the ribosomes, which look a lot like little dots, and the ribosomes will build the protein. So we have DNA, that's our instructions inside the nucleus, our mRNA, which can read little sets of those instructions, read your genes, so to speak, and then take those instructions out of the nucleus to the ribosomes, and then the ribosomes will actually build protein, which we're going to talk about a little bit. But the ribosomes themselves, what makes the ribosomes? Well, inside your nucleus, you have this dark area called the nucleolus. Sounds almost like nucleus, but nucleolus. And the nucleolus builds the ribosomes. All right, so that's inside the nucleus. So here's a picture. So here's your cell. This is the nucleus right here. Notice it does have these little pores that allow certain things to go in and out. So the nucleus has a membrane and it has pores. This is a nucleolus, this dark area inside the nucleus. And all of this would be the actual DNA. which would be in a mass, notice they wrote here, called chromatin or chromatin. And that mass, the chromatin, is actually your chromosomes, but you only see them as chromosomes. You may remember, maybe learning a little bit about cell division, how the chromosomes look like these little X's. They only look like that when the cell's dividing. The rest of the time, they're sort of uncoiled into this stringy stuff called chromatin. Okay, so let's talk about the ribosomes. So the ribosomes are made out of a special kind of RNA called ribosomal RNA and proteins. So they're actually made of two things. And again, their job is to build protein from the instructions of the DNA. And any kind of cell that's making lots of protein, like, for example, cells in your body that might be making hormones like insulin or things like that, or cells that are making enzymes that your body needs, they're going to actually have a lot of ribosomes because they're building a lot of protein. So again, going back to our original picture, we've got our nucleus, our DNA is in there, in that mass called chromatin. Our nucleolus, which doesn't actually have a membrane around it, it's just a dark area. The nucleolus makes these ribosomes, and the ribosomes they look a lot like little dots. They end up outside the nucleus. A lot of them are lining something called the rough ER. That's why it's called rough, because it actually looks bumpy under a microscope. So you'd see the ribosomes here. But some of the ribosomes are actually just floating around in the cell as well. So you have some ribosomes that are attached to endoplasmic reticulum and some that are just floating around. Both of them make proteins. Usually the difference is that the ones that are floating around are making proteins for inside the cell, like proteins that the cell needs to keep itself alive. And the ones that are along the rough ER, they're usually making proteins that are eventually going to get exported from the cell. So a cell might be exporting, like I said, like an enzyme or a hormone or something that the rest of the cells in your body need. And so ribosomes that make those would be along this tubular stuff, which is called the rough ER. And we'll talk about that in a minute. Okay, moving on. This is a picture showing ribosomes here, some that are freely floating. And then notice this is what I was telling you is called the rough ER, or the rough endoplasmic reticulum. And see the dots, the ribosomes that are lining that. And a ribosome, even though we always draw them as dots, by the way, they're not actually dots. If you were to magnify them enough, they look like this. They're actually made out of a big piece called the large subunit and a little piece called the small subunit. And they actually build proteins. mRNA, remember DNA was the instructions. mRNA is a special kind of nucleic acid that reads those instructions, brings them out to the ribosome, and then the ribosome will actually build the proteins. So your ribosomes are the workers that are going to put the proteins together that your cell needs or might need to export for some other job. Now, the rest of the membranes in your cell are part of something called the endomembrane system. These are organelles in your cell that work together. So they might build things, distribute things, store things, or export things from cells. So let me show you the parts of the endomembrane system. So the first part is the ER, or endoplasmic reticulum. And there are two types of endoplasmic reticulum. So smooth ER, smooth endoplasmic reticulum, it is responsible for making lipids. It also makes steroids. Remember, steroid hormones were a type of lipid. So steroids are made by your smooth ER. And it also processes toxins. So smooth ER would be responsible for detoxifying substances that you eat, because everything you eat has things in it that your body may need to take out. Chocolate, for example, has chemicals your body needs to break down. Think about coffee and sodas. Caffeine has to be broken down. Antibiotics, if you're on them, they have to be broken down. Any kind of medications you take. And so this would process those things, the smooth ER. You have a lot of this in your liver because that happens to be an organ in your body where you detoxify a lot of things after you digest and absorb your food. The rough ER, it specifically makes proteins. It has, it can also make, it mentions that it makes membranes, but remember this is the one, it's called rough because it looks bumpy, and it specifically makes proteins that will become part of the cell membrane and proteins that are going to be exported. The ribosomes actually build the proteins. What it really does is it kind of It's kind of not clear here, but I always like to explain that it modifies proteins, that the ribosomes make them, and then the rough ER modifies them. So it's a biosynthetic workshop because it's putting all kinds of finishing touches. Number one, it's building steroids and lipids. Number two, it's detoxifying things. Number three, it's modifying and putting the finishing touches on proteins that the ribosomes made. And so it's building all kinds of molecules that your cells need or maybe need to export for other cells to use. Here's a picture. So the rough ER looks like this. It's got a little dots around it. That's the ribosomes. That's why it's called rough. And then the smooth ER doesn't really have the ribosomes around it. They're usually located and attached to the nucleus. In fact, that's why this is called the endomembrane system, because the nucleus has a membrane, and the membrane of the nucleus is actually attached to the rough and smooth endoplasmic reticulum. So this is just showing, and then what will happen is when the ribosomes make a protein, the protein goes into the rough ER. This is the rough ER. Notice in this case, the rough ER is modifying that protein. In this case, it's adding little sugars to it to make something called a glycoprotein. And then it's not like the protein just leaves. What will actually happen is pieces of this rough ER will break off and carry this product to the next place in the cell. So you have this rough ER, and when the protein is finished, it literally buds off a little membrane-bound vesicle, and that's going to go to the next location. So again, this is why this is called the endomembrane system, because pieces of cell membrane, the membrane around this, is breaking off and traveling to another place. The nuclear membrane is attached to this, and then pieces of this can break off and become attached to the next part, which is the Golgi apparatus. So the Golgi apparatus looks like a little stack of sacks. And what it's going to do is it's going to take all the products that the endoplasmic reticulum made, finishes processing them, and then can send them to the cell surface, the cell membrane, or even export them from the cell altogether. So this is what it looks like. So notice the ER was sort of long folded canals. The Golgi bodies don't look like that. They're like a little short stack of pancakes, no ribosomes around them. And what will happen is, here's the vesicle. This is coming from the endoplasmic reticulum. It broke off, and now it fuses with the Golgi apparatus, or Golgi bodies. Sometimes it's also called the Golgi complex. It has a couple of names. And it dumps the contents off here, where they'll get modified some more and packed up. I like to think of it as like the UPS. And then another vesicle will form. This literally breaks off and takes this to the cell membrane, where it might become part of the cell membrane, or it might get completely exported from the cell. So to summarize, just to show you here, I'm going to go back to this slide so there's a blank space. So if a cell was going to make a protein, here's all the steps together that would have to happen. So the DNA in the nucleus would have a code. That code would be read by mRNA, which is a messenger, and it would carry that message out to the ribosomes. The ribosomes would build the protein. And then, here's your rough ER. Right from the ribosomes, this product would go into the rough ER, and this protein would get modified. Maybe it would have sugars added to it. Maybe it would be cut or be folded into a specific shape. And then after that happens, this pieces of this rough ER will bud off, literally bud off, and take this protein now that's been modified to our next stop, the Golgi apparatus. These will actually fuse with the Golgi apparatus, dump off this product where it might get further modified. And then finally, little buds will come off of this and fuse with the cell membrane like this and dump these contents out. So maybe this was a hormone that your body needs, and this will get dumped out into your bloodstream where it can be carried to another location. So that's called the endomembrane system, because you actually have little pieces of the membranes of these different organelles that break off and fuse with other organelles to work together to make proteins and lipids for your body.